We're in a rapid growth phase at the moment where we've been growing through capabilities within our labs and also headcount as well. And that's really been driven by an increased demand for our technology. We're expecting a lot more of that growth over the next couple of years, and so we're expecting a lot more relationships with these pharmaceutical companies and these synthetic biology companies that we're working with. There were kind of three major problems that we set out to solve as a company. One is that we recognised that the existing technology for making synthetic DNA was very inaccurate. We would have many problems in the lab with DNA that was coming in where it would contaminate downstream reactions. By extension of that, there is more of a pressure on the field of DNA synthesis where we need ever increasingly longer pieces of DNA, really thinking about making gene length pieces of DNA. And so the problem there is that, by extension, is that there are many genes that are very, very difficult to make. And so that was another problem that Camena tried to set out to solve. The third component that we've spent a lot of time developing is really to think about sustainability. The existing technology that's really been the gold standard since the 1980s uses a huge amount of solvents and generates a huge amount of solvent waste. And Camena is really developing a sustainable version of that of DNA synthesis technology.
We're here in one of our two labs. This is where most of the gene synthesis process happens. And our gene synthesis process starts by taking our long target gene and in silico fragmenting that up with our proprietary algorithm into much smaller fragments that we start the build with. Our process starts with small building blocks, which we have in these 96 well plates. They're then used to assemble our fragments of the gene of interest. So once we have our building blocks and our smaller fragments, we then go through a series of enzymatic steps to join those together, where the enzymes will chop up the DNA and then allow them to join together, and we do that in one of these machines. So once the enzymatic steps are complete and we have our larger fragment of the gene, we can then put those into the bacteria, grow those up, and then go through multiple rounds of this process to get our full, complete gene. So as you can see, we use blue-white screening to ensure we pick the right colonies, and from that we can extract our full gene of interest, ready for delivery to a customer.
So our technology is really being used at the moment predominantly with customers that are very high consumers of genes. Many of those are on the pharmaceutical side, so very well established pharmaceutical companies that are, as part of their research pipelines, are consuming huge numbers of genes. The problem, or perhaps the market, if you like, for synthetic DNA is a multi-billion-dollar industry. And that covers a range of industries from agri-tech, diagnostics, vaccine production, for example, which the public are very familiar with COVID, to more discovery of antibody and other therapeutics. So it's a huge market across the world. Yeah. We set out to first of all develop intellectual property, really to protect our technology. And then we got to a point where we felt like we developed the technology to a sufficient position where we could then start to sell genes. We then set out to find institutional investors to support that growth and really to grow very rapidly, moving away from the previously organic growth of the company. And so Camena Bioscience is a UK-based company. Specifically, we're Cambridge headquartered company, and we were really looking for someone with life science experience, but also to be a UK-based fund that would support us on that growth, and that's why we found that Mercia really fit the bill. This year we've expanded quite a lot. We're growing from roughly about 5 to 20 people. We're roughly about 18 at the moment, and in addition to that, we're also developing capabilities within our laboratories and our production facilities, which is through forms of automation and the equipment and liquid handling that's allow us to really increase our throughput. We're what people might refer to as kind of like a core tool, really enabling scientists and other biotechnology companies to kind of fulfill some of their aims and objectives. And so through our improved technology, they're now able to do things that previously they weren't able to. So they get access to genes they previously won't be able to access, and so that means that they can begin to start developing potentially new therapeutics, discover new enzymes, new antibodies, new gene therapies. And so we're enabling them on their side.